Okay, good afternoon. It's still 1 minute to 5, but I think we can start. And today it's my big pleasure to welcome Professor Emmanuelle Charpentier, who accepted the invitation from the MENDEL Lectures Committee and came to Brno to give her MENDEL lecture talk. And I guess most of you are not microbiologists, but probably most of you are affected by the results of her work. So I would like to give you a short overview of Emmanuelle's professional life. So she's got her bachelor, master's, and a PhD degree at the Institut Pasteur and the University of Pierre and Marie Curie in France, in Paris. Then went for several postdocs, mainly in the United States, at the St.Jude Children's Hospital, Rockefeller University, New York University Medical School, and Skirball Institute in New York. And then she accepted a position at the University of Vienna, where she has spent 7 years, and then moved to University of Umeå in the northern Sweden, where she has stayed for another 5 years. It wasn't clear. And at the same time, or later, during her Umeå season, she also accepted a position at the Max Planck Institute for Infectious Diseases in Berlin. And it's a long and successful life. So, and then in 2018, she became a founding and the acting director of the Max Planck Unit for the Science of Pathogens, and is affiliated also as the honorary professor to the Humboldt University in Berlin. So she came to visit us from Berlin. And you probably know us already from the title. What's her biggest scientific success that I believe is, and I'm not the only one, believes is a breakthrough that makes our lives as the life scientists or cellular biologists or microbiologists or plant biologists, whatever biologists, much easier. And it is the discovery of the CRISPR-Cas9 system and its application in the genome editing. And Emmanuelle has been awarded numerous prizes for this achievement, including the Breakthrough Prize in 2015, and she's been also among in the most general public. She was ranked several times among the 50 or 100 the most influential people in the world, depending on the magazine. And I would like to, before I give the microphone to Professor Charpentier, I would like to pass her, on behalf of the Masaryk University and the MENDEL Lectures Committee, the medal. And this is the medal of Gregor Johann Mendel for the outstanding achievements of science. Thank you.
Thank you very much. It's very nice. Thank you. Thank you very much for the kind introduction. My life has not been always successful, but you don't have success if you don't have failures. And I think I had a number of failures in my life, but a number of successes as well, so then it's very good. Having said this, I would like to thank very much the committee for having selected me as a speaker of the MENDEL lecture. I will try to not be too long, but I'm afraid that I'm going to give a long talk, and because I got to know that the lecture will be attended mostly by PhD students, therefore I will enter quite much in the details of how we came to CRISPR-Cas9. I could talk about CRISPR-Cas9 for days, but I will try to keep it to 45 minutes or maybe a little bit longer. So my laboratory is right now located in Berlin, and at the beginning of last year I have founded a new small institute, Max Planck Institute, that is called Max Planck Unit for the Science of Pathogens, so really focusing on microbial research. Since I'm a microbiologist, I have been mainly in my life a microbiologist, and I want to support this field further, specifically because you will understand that at least it came to the very nice technology CRISPR-Cas9. I will start with some acknowledgements. So this research started when I was in Vienna, at the University of Vienna, Max F. Peretz Laboratories. The research about CRISPR-Cas9 was mainly done during my time in Sweden at the Laboratory for Molecular Infection Medicine Sweden at Umeå University, and that is a Nordic, has a Nordic partnership with EMBL, the European Molecular Biology Laboratory, mainly based in Heidelberg. And then I moved a couple of years ago to Germany, first at the Helmholtz Center for Infection Research in Braunschweig, and then in Berlin at the Max Planck. I should say that my time in Sweden was very helpful. I just refer to maybe the time of MENDEL here at this place. You may imagine the north of Sweden as a very quiet place where you can hide easily and focus easily on science. And for me, it has been a time that has been very much rewarding. You know, people understand that you stay focused on your research and no one bothers you. So it's very nice. Having said this, here are also the acknowledgements of the people involved. First, very young researchers in my team, and it's on the bottom left. And I want to thank specifically Krzysztof Szelinski and Elica Delcheva. So the work was actually started by a postdoc and 2 PhD students and a master's student. But the main story started really with Elica Delcheva, a master's student from Bulgaria, Krzysztof Szelinski, a PhD student from Poland, and then other people in my lab who followed up. There was a first collaboration with a group of Jörg Vogel in Germany with Cynthia Sharma, a second collaboration with a group of Jennifer Doudna in the US with Martin Jinek, coming from Czech Republic, and collaboration with a group of Eugenie Kounin, actually originally from Russia, but based in the US. So this was a story that developed quite much with young scientists originating from Poland, Bulgaria, and Czech Republic. I wanted to mention this. It's important. I'm in Czech Republic now. So I want to thank all those people who have helped developing this project. Here, I start by showing you the first page of an article that was published in Science nearly 6 years ago now, 5.5 years ago, entitled The CRISPR Craze, with a subtitle, A Bacterial Immune System Yields a Potentially Revolutionary Genome Editing Technique. So it's about this technology that allows precise gene surgery in any cell and organism. So this article was published only a year after we published a paper in Science in 2012, describing the last step of the CRISPR-Cas9 mechanism and explaining how this last step of the mechanism could be harnessed as a powerful technology that could be useful to biologists in different areas of life sciences. And as a matter of fact, a year later, this was clear that this technology was very successful. At the beginning of 2013, so a little bit more than 6 months after we described the technology and provided guidelines how to use the technology, a number of articles started to be published with regard to the application of the technology to perform precise genetics in human cells, in mice, in plants, and in other model organisms that are used to understand the mechanisms of life. So this technology, most of you may know, how the technology works, but for those who may not be that familiar. So this is the target DNA, so it's really a technology that targets the DNA, and it's composed of an enzyme called Cas9 that is here represented as blue scissors. And this enzyme Cas9 is guided to a specific sequence of DNA of interest by an RNA molecule that works in a sense like a GPS system that will recognize a certain sequence on the DNA and will bring the enzyme to cleave the DNA at a specific location. So this programmable RNA is here represented as a single guide RNA, and in nature it's a duplex of two independent RNA molecules, so the CRISPR RNA and the tracer RNA. And there is another component that has to be present on the DNA to be cleaved. It's a yellow part here representing the protospacer adjacent motif called PAM, and it's a short stretch of nucleotides. In the case of the CRISPR-Cas9 system that is used worldwide and that is originating from the bacterium Streptococcus pyogenes, this PAM sequence is composed of a short stretch of nucleotides, N for any base and GG. And so this PAM has to be located upstream of the sequence, and that will be cleaved by Cas9. So the scientist just needs to design the RNA molecule to have this portion of the RNA molecule complementary to the sequence to be cleaved that is located upstream of the PAM sequence. So it's a mechanism that is looking simple. Indeed, it's as simple as it is shown here on the picture, but yet sophisticated enough so such that the technology has been developed extremely fast in multiple versions and in multiple tools in a way that are composing now the CRISPR-Cas9 toolbox, and all those tools allow not only to modify the DNA but also to modify the expression of the DNA, and I will give some examples later on. Before I start with CRISPR-Cas9, I would like to place this discovery in the perspective of the genetics that has been going on and studied since the beginning of the 19th century with fundamental genetics, and that was established mainly with regard to the rules of fundamental genetics in the 19th century with Darwin on the origin of species, Mendel, we are here, and I had the chance to visit the museum that is extremely inspiring. So Mendel on the laws of segregation of alleles, and Friedrich Miescher, who isolated the DNA in 1871. One needed to wait 80 years to have the discovery that DNA was the carrier of genetic information. And later on, the structure of the double helix of DNA that was determined. And in 1966, the genetic code that was revealed. And surely the need to understand the genetic code, the functions of genes, and understand genomes in general. And here came the era of genetic technologies with the basics that were established, so starting in the 60s, but really more in the mid-70s with the restriction enzymes and different types of enzymes that would allow to recombine the DNA, and also later on the possibility to sequence the DNA and to amplify the DNA. So these are just some milestones. Surely later on, RNA interference came into play, and the possibility to modulate gene expression using small regulatory RNAs. And so these technologies became critical and allowed to start the field of molecular biology, and became critical to advance all the knowledge that we have now with regard to mechanisms existing in life sciences. But what is important to remember at this stage is that all these discoveries of enzymes were mainly based on research done on bacteria and viruses. Here I would like to cite Jacques Monod, a French microbiologist, who was actually working and got awarded the Nobel Prize on his work together with François Jacob and André Wolf. So focusing on bacteria and bacteriophages, the viruses of bacteria, focusing on proteins with enzymatic function, focusing as well on genes organized in operons, and on the hypothesis that RNAs could also carry regulatory function. So I just cite this because all those different, criteria, features of biological mechanisms are really what you find in CRISPR-Cas. So it's all about proteins having enzymatic functions, RNAs, operons of genes, catalytic functions, and also how bacteria defend themselves against bacteriophages. But he had an interesting quote, which he mentioned in a book that he wrote in 1970, Chance and Necessity. Modern molecular genetics offers us no means whatsoever for acting upon the ancestral heritage so as to improve it with new features, to create a genetic superman. On the contrary, it reveals the vanity of any such hope. The genome's macroscopic proportions today and probably forever rule out manipulation of this sort. So I found this quote interesting because I'm sure that now we will be most likely surprised to see the power of the CRISPR-Cas9 technology, and I'm also curious to know what what Mendel could have thought of, where his discoveries would have, in which direction his discoveries would have evolved in the future years and centuries. Having said this, François Jacob also had a very interesting quote that was, nature is a tinkerer, not an inventor. And it's an interesting quote because it's all about, also it refers to CRISPR-Cas9 as a mechanism that does some tinkering on the DNA. It's really a natural mechanism that does some bricolage on the DNA. And we are here to study those mechanisms and are happy when we find an interesting mechanism that can be harnessed further. Having said this, CRISPR-Cas9 is really belonging to what we call gene editing. So the possibility to edit genes and genomes in a more precise manner. And prior to CRISPR-Cas9 in the field of gene editing, there were two types of nucleases that came to be engineered really for exactly the same purpose than CRISPR-Cas9. These are the zinc finger nucleases and talon nucleases. So these are artificial nucleases that take into account for sure properties of natural enzymes, but they have been engineered to really perform exactly what CRISPR-Cas9 is doing. However, the issue with those nucleases is that one needs to engineer a new nuclease for any new type of DNA to modify, because the code, the programmability of these nucleases, allowing them to recognize a certain sequence of the DNA of interest and cleavage is contained within the proteins. So those nucleases were used, commercialized, not very much democratized because of the cost of these technologies, and also the fact that it's quite cumbersome to always have to produce a new protein. So there was really a need for a fast and easy tool, and this is what CRISPR-Cas9 brings, specifically thanks to the programmability via the RNA component, whereby there is one enzyme, but the biologist just needs to synthesize and to design specifically this RNA molecule. So the research in my lab with regard to CRISPR-Cas9 came from our interest in understanding how bacteria as human pathogens can attack the human host. To a certain extent, we try also to understand how the human host defends itself against bacteria. We focus mainly on Streptococcus pyogenes, that is the main source of the CRISPR-Cas9 technology that is used worldwide. Streptococcus pyogenes, it's also called group A Streptococcus. It's a human pathogen. It's a strict human pathogen. It infects only human cells. It's not found in another environment. Here you can see the cocci of the bacteria, so forming cocci that are represented in red and that are invading human pharyngeal epithelial cells that are here colored in green for the purpose of the picture. So this pathogen is responsible for a wide range of diseases, from mild infections such as pharyngitis or tonsillitis to more severe infections such as toxic shock syndrome or necrotizing fasciitis. We are mainly interested in understanding how bacteria can adapt to their environment in the human host, whereby they are encountering a number of stresses and whereby they have to have different regulatory mechanisms in place that are expressed in spatial and temporal manner, and that are involving molecules of protein nature and RNA nature to ultimately lead to a modulation of expression of different factors that are important for the virulence or for maintaining the fitness of the bacterium. This project on CRISPR-Cas9 really came from our interest in working on small RNA molecules. When we started to work on small RNA molecules, the knowledge was as follows. So for sure, DNA is transcribed into RNA, and the RNA is translated into proteins. You have also these transfer RNAs and ribosomal RNAs involved in the translation of proteins. And you have the small regulatory RNAs that actually were discovered first in bacteria in the control of replication of plasmids, and ultimately were found to also interact with the expression of messenger RNA and also have roles as such in virulence or in different aspects of the metabolism or fitness of the bacteria. So small RNAs can interfere with messenger RNA, forming duplexes of RNAs, and as such either affect the stability of the messenger RNA or interfere with the translation of the messenger RNA. So for sure you have different types of mechanisms that have been described in this regard. And they can also interact directly with proteins to activate or sequester them from their functions. So in bacteria they are called small RNAs or small non-coding RNAs. In eukaryotes they are called microRNAs or small interfering RNAs or other types of RNAs that have been discovered since. What was not described when we started to work on small regulatory RNAs at the beginning of 2000s were small RNAs that will interact directly with the DNA and change the fate of the DNA. So this is what CRISPR brings. Having said this, in 2006-2007, so a bit more than 10 years ago, we started in my lab to investigate small RNAs in Streptococcus pyogenes. So we were already working on small RNAs for which we could find a role in virulence, but we were interested at that point to identify more small RNAs. So we used the bioinformatic approach, and here is represented the genome of Streptococcus pyogenes that is relatively small, and highlighted the different small RNAs for which we could find either a function analogous to function that were already described for housekeeping small RNAs, and other small RNAs that seem to be new. We looked at their expression by Northern blot analysis, and we focused on one RNA, which ultimately is now known as tracrRNA. We could find a nice expression of this RNA. So this is a Northern blot analysis of the RNA, of the tracrRNA expression in Streptococcus pyogenes, grown in vitro in test tube from early log phase to late stationary phase. So it's an RNA that is expressed in 3 forms and expressed in a quite constitutive manner. So this is a loading control. We mapped the 5' end and the 3' end of the RNA by different techniques, including primer extension. And what was clear already when we started to work on this RNA is that this RNA was encoded by a piece of DNA located upstream of a gene that was highlighted at the time in 2007 as a gene encoding a protein that will be a CRISPR system-related protein of Streptococcus thermophilus, another streptococcal species, and that this protein was already how do you say, hypothesized to have two nuclease domains, so to have the Russell-like nuclease domain and the HNH-like nuclease domain. So this is from the work of bioinformaticians, I will discuss this later, who could identify the number of CRISPR-related proteins having these domains, in this case here, nuclease domains that would be involved in cleaving the DNA. Having said this, we started to focus on this tracer RNA, and this led us therefore to this CRISPR system as well, because of the location of tracer RNA, but also because there were two reasons to work on those two RNAs. First of all, the CRISPR field at the beginning of 2006, 2007 was already starting with some history that I will describe in the following slides, but also because we started to work on tracer RNA independently of CRISPR RNAs. The idea with tracer RNA was to find a target, to be able to describe the function of this tracer RNA. And initially, we found as a target a messenger RNA. So we could show that tracer RNA had a very nice interaction with a messenger RNA coding for a virulence factor in Streptococcus pyogenes. So we are very happy because we thought that we could attribute to this RNA a function of regulatory RNA in the virulence of Streptococcus pyogenes. However, we could not make any sense of this interaction, even though later on in another bacterial species, it was shown that tracer RNA can interact with messenger RNA coding for virulence factors. Having said this, bit by disappointment, I decided to see whether, and to ask the question whether tracer RNA would not have a function on the CRISPR system. So coming with CRISPR is a defense system existing in bacteria and archaea, and it brings me to another type of infection, where the bacteria are not the pathogen attacking a human host or another host. Bacteria are hosts themselves, and they can be attacked by mobile genetic elements, and they have surely evolved different defense systems to counteract this attack. So among the mobile genetic elements, you have the plasmids, the transposons, and the phages. So all these mobile genetic elements can bring new genes to the bacteria and contribute to bringing to the bacteria new features, such as antibiotic resistance or virulence functions or functions of adaptability to the environment. With regard to the phages, so the viruses of bacteria, once a phage injects its DNA into the bacterial cell, the phage can enter a lytic cycle, whereby the DNA is going under replication. You have production of phage particles that will lyse the cell and will propagate to lyse further cells. The invading DNA can also, of the phage, can also, for some bacteriophages, integrate as a form of prophage into the genome of the bacteria. So this is what we call temperate phages. So they enter the lysogenic cycle, and in this case, they really bring new genetic features to the bacteria, and for sure this is propagated upon cell division and to the population. Having said this, so those mobile genetic elements can also attack archaea, and archaea and bacteria have evolved different defense systems. For example, as a defense system that is regarded as an innate immune system, you have restriction modification. With a restriction modification enzymes that can recognize the DNA of mobile genetic elements and cleave this DNA. All the systems, defense systems that were described before to CRISPR-Cas9 were labeled as innate immune systems because they were not involving a first stage of adaptation and recognition. CRISPR-Cas is the first and so far only adaptive immune system that is known in bacteria and archaea. Having said this, I would like to mention that it's not present in all bacteria and archaea. It's more present in archaea, actually. It's present in 90% of the archaea, but only 50% of the bacteria because you have a diversity of defense system and it's just another accessory defense system. It's more present in the archaea because obviously archaea, in their drastic environment in which they live, they are more prone to viral infection. Having said this, with regard to the history of CRISPR-Cas, this is something I wanted to mention briefly. In 1987, so more than 30 years ago, a Japanese group made the observation of short repeats on the genome of Escherichia coli. At the late 90s, what was shown is that the CRISPR array was transcribed and the transcript was obviously processed, so they could show this by Northern blot analysis. If they would look for the expression of these repeats, they will see different sizes of RNAs. So with the idea that those repeats will be transcribed as a long precursor CRISPR-RNA molecule that will undergo processing to lead to this to shorter forms of RNAs. Then in the beginning of 2000, surely bioinformaticians started to look into those repeats and found these repeats in more bacteria than Escherichia coli. And what they found as well is that the repeats, this series of repeats were located in the vicinity of genes that were organized as an operon, and genes that were encoding for what they called later Cas proteins, for CRISPR-associated proteins, and proteins that would have as characteristics to contain domains that would be domains characteristics for proteins associated with DNA or RNA, or proteins that can cleave DNA or RNA. Then what was also described in 2005 is that interspaced in between those short repeats that are short sequences identical to one another, they could find that the sequences that were interspaced in between the repeats were unique on the genome and were originating from mobile genetic elements, so meaning from phages or plasmids. And so they came or even transposons as well, but mostly phages. So they came to the idea with regard to former experiments showing that these repeats were transcribed into RNA molecules and that there were these proteins with these properties which I have described, that CRISPR-Cas will be an RNA-based prokaryotic immune system, and that there will be a memorization of the infection involved, and that these sequences that are represented here in different colors are somehow memorizing a memorization of former infections with mobile genetic elements. So in 2007, there was an article published in the journal Science by the group of Philippe Horvath and Sylvain Moineau, showing that indeed CRISPR-Cas is an adaptive immune system. So they did simple experiments using Streptococcus thermophilus that is used for yogurt production, whereby they actually ultimately used this technology CRISPR-Cas9 in a natural fashion to produce Streptococcus thermophilus strains that will be resistant to phages used during the production of yogurts. But in principle, the idea is that you have a first infection whereby the phage injects its DNA into the bacterial cell. There will be CRISPR-Cas proteins involved that will recognize the phage DNA upon infection, cleave a portion of the phage DNA and insert it at the level of the CRISPR array, so at the leader sequence. And this sequence will memorized, will then be transcribed into RNA molecules, so the CRISPR RNAs that will be processed, associate with proteins, and allow the immunity upon reinfection by the phage. So in 2008, the group of Stan Browns and John van der Oost could show that for one type of CRISPR-Cas system, that is not CRISPR-Cas9 but another type, there were different proteins, Cas proteins, involved for the recognition of the phage upon a second infection. So this is what we call interference, and whereby there will be different proteins forming a complex that will be guided by a CRISPR RNA molecule that contains the sequence of the phage that will then be recognized. There is a duplex formation between the so-called spacer sequence that is in color of the CRISPR RNA with the so-called protospacer sequence of the phage that infects a second time the bacterium, and one protein will recognize sequence specifically this duplex and will cleave the DNA, and thereby the DNA cleaved cannot be maintained for replication, and this is loss of the maintenance of the phage. In 2008 as well, the group of Santheimer with Marafini, shown in the paper that was published in Science, that another type of CRISPR-Cas system, another type than this system, would indeed recognize the DNA. So this will be the DNA that will be recognized. I just mentioned this because initially the bioinformaticians hypothesized that with regard to the fact that CRISPR-Cas will be an RNA-based prokaryotic immune system, they made an analogy with the RNA interference system existing in eukaryotes, but this RNA interference system in eukaryotes target messenger RNAs. And so it was not so clear whether CRISPR-Cas will target RNA or DNA, even though they had the suspicion that it will target DNA based on the homology of Cas proteins involved with proteins cleaving DNA. So this leads to the adaptive immune system, so whereby you have a first phase of recognition with acquisition of a so-called spacer, so sequence of the phage, then transcription of this memorized mobile genetic elements into a long RNA molecule with processing mature CRISPR RNAs that each can associate with a complex of CRISPR-associated protein, and then the CRISPR RNA will guide this complex of CRISPR-associated protein to the invading DNA and cleave it. So this was the knowledge that was published during the course of the start of the CRISPR-Cas9 project in my lab. What was important to mention is that everyone was working on type 1 and type 3 CRISPR-Cas systems, and here is just a slide showing you roughly how the system CRISPR-Cas has evolved in bacterial archaea. So there are two classes that have been described up to now with 6 types and further subtypes, and you have a lot of these further subtypes and a large diversity of these CRISPR-Cas systems in bacterial archaea. The common feature is really, in principle, the different, the immune, I would say, cycle that is conserved. Another conserved functions are the Cas1 and Cas2 proteins that are important for the stage of recognition, specifically allowing the integration of DNA of the phage upon the first infection into the CRISPR array. And all those proteins here that you can see is really showing you the diversity of the CRISPR-Cas protein. So you always have the CRISPR array. The repeat sequences are evolving tremendously. Surely you have always a diversity of mobile genetic element sequences in between the repeats. There are always some Cas genes, so always an RNA components involved and CRISPR-associated proteins involved, but the CRISPR-associated proteins in terms of their nature are diverse, and this is mainly the basis of this classification. So when we started to work on CRISPR-Cas9, and we started in 2007, 2008, so there were all these papers coming up showing mainly for type 1 and type 3 that there was a complex of CRISPR-associated proteins involved at the level of interference. For sure, us in Streptococcus pyogenes, we were lucky enough to have to work with a strain that contained two types, one type, subtype 1, and one type that was described later on to be type 2, with a Cas9 protein that consists of, in principle, one single protein for obviously for the stage of interference, and this was shown in the paper in 2007 by the group of Sylvain Moineau and Philippe Horvaths, that there will be this protein that most likely will be involved, even though the mechanism will not be known at that time. This protein had different names. It was called Cas5, CSX12. We started to work on this protein. The name was CSN1, and it switched to Cas9 name starting 2011, 2012. So in principle, you see the difference already is that here you have one protein involved. And another also characteristic is that what was shown for type 1 and type 3 is that for the processing of the CRISPR RNAs, there was one CRISPR-associated protein involved, the Cas6, that works as an endoribonuclease. However, the Cas6 was absent for the type 2 system. So we knew that we would be, studying a totally different mechanism. So this motivated us to continue, and specifically with this idea to look whether tracer RNA will not have a function in the CRISPR-Cas9 system. So this was not expected because CRISPR was supposed to work sufficiently with the CRISPR RNAs and the CRISPR-associated protein. But with the type 2 system, it's obviously different. So to make a very long story short, of very classical approach of bacterial genetics and bacterial biochemistry, we came to the understanding of the CRISPR-Cas9 mechanism. I just say this because these are really experiments that could have been done 30 years ago, even 40 years ago. However, what was really critical in nevertheless understanding better the interaction, or let's say not really the interaction, but to somehow give us more proof that tracer RNA was a very well expressed tracer RNA in the cell. And these were differential RNA sequencing analysis that allowed us to come to the point that tracer RNA was abundantly expressed in Streptococcus pyogenes, but that also allowed us to define how in vivo those RNAs were processed. So in the type 2 system, the way it works is as follows. We could identify that there is indeed one protein, Cas9, so one single protein involved in the process of interference, whereby you have a first process that is leading to maturation of the CRISPR RNAs, so whereby the long precursor CRISPR RNA is transcribed with the different mobile genetic element marks. Tracer RNA is transcribed. What we could find is that tracer RNA has an anti-repeat sequence, and that allows it to base pair with each of the repeats of the long precursor CRISPR RNA, forming duplexes that will be recognized by the enzyme RNA3 from the bacterium. So it's different from the type 1 and type 3 system that are using Cas9 proteins to cleave the CRISPR RNA. So here it's done by the endoribonuclease 3 from the bacterium with Cas9 that has a role to stabilize the duplex of RNA. And then upon further maturation with another type of endoribonuclease or exoribonuclease, which we still did not identify, you have so really the mature complex that is composed of this duplex of RNA that is stabilized by the protein Cas9. And this is really the interference complex. So initially what was not clear is whether tracer RNA at this level will leave the system, so it will be only necessary to cleave the CRISPR RNA and allow its maturation. But in the lab we knew that tracer RNA had the property to remain bound to Cas9, and this was really the reason why also Cas9 at the beginning was recognizing the duplex of RNA, and there will be maturation going on, and at the end of the day, a very unique enzyme that is bound to a duplex of RNA molecules. And this enzyme ultimately, guided by this duplex of RNA, can recognize site-specifically the DNA of the cognate phage. And so here you have first the protein Cas9 that will recognize the PAM, the protospacer adjacent motif, that is for the case of Streptococcus pyogenes, a short stretch of nucleotide, as I said at the beginning of my talk, NGG. Once the protein, after the protein has really looked for the PAM sequence, it will look further for a complementarity between the CRISPR RNA and the target DNA. There will be R-loop formation and then always constant changes of the positioning and the structure of the complex, so that the two nuclease domains can be well exposed to allow the enzyme to cleave each strand of DNA using each of the nuclease domain, HNH and ROC motif. And the enzyme has the ability to cleave by introducing a DNA double-stranded break, so in a blunt fashion, 3 base pairs upstream of the PAM sequence. So for sure, this was a very nice result, and when my student Krzysztof Szylinski did the experiment in my lab, it was working perfectly at first. And so what came after is the idea of linking those two RNA molecules together to have a single guide RNA. And what we could show in collaboration with a group of Jennifer Dorna with Martin Jinek is that indeed, as this single guide RNA was working very well, like the natural duplex of CRISPR RNAs and tracer RNAs to cleave sequence specifically a piece of DNA, and the ability to program this RNA as a technology to cleave any piece of DNA of interest that will be located upstream of this NGG. So here you see more details of the cleavage, whereby you have the CRISPR RNA molecule, the tracer RNA that forms a duplex thanks to its anti-repeat sequence with the CRISPR RNA. Here you have the spacer sequence of the CRISPR RNA that base pairs with the target DNA, and the cleavage that occurs 3 base pairs upstream of the PAM. And this was in accordance to a cleavage that had been observed by the group of Sylvain Moineau in vivo. And this was a paper published in Nature in 2010, where he showed that indeed Cas9 was able to cleave in vivo 3 base pairs upstream of the PAM. However, at that time, this was genetic experiments that were performed, and tracer RNA was not mentioned in this paper because it was not identified at that time. So what CRISPR-Cas9 does is that it does exactly what thin finger nucleases and talon nucleases can do. In a programmable fashion, it introduces cleavage on the DNA in a blunt fashion. And once the system has cleaved the DNA, the rest is done by the cells. And in eukaryotic cells, you have triggering of different types of repair mechanisms. So depending on the type of the cells, you have one mechanism working better than the other one. And surely it depends also how you trick the system. If, for example, you just cleave the DNA, you can have non-homologous enjoining repair mechanisms that are triggered and that will allow to introduce smaller or larger indels, so deletions at the site of the break. And if you bring to the cell an homology donor, you can have a replacement of a piece of DNA by another piece of DNA, and by this you can introduce new sequences on the DNA. You can perform knockouts, so will it delete a portion of the DNA. You can also introduce specific mutations or correct specific mutations. And so the technology has been shown to be, for sure, extremely powerful. The hypothesis was that if Cas9 was working very well, which actually personally I thought it would, that it will be very powerful and that it will allow to provide benefits for genome engineering in all three domains of life, and that it will have applications in life sciences, biotechnology, agriculture, and medicine. What is important to say is that the technology, it's relatively cheap and easy, for sure. It may not apply for the technologies that are necessary after to verify that the modification has been done in a precise manner, because this involves a deep sequencing analysis that is often costly for some labs. The technology is described as being efficient, certainly versatile, and I will show you some examples. Also allowing multiplexing, in the sense that some have shown that the technology can be used at once with different guide RNAs that target different locations on the genome to perform different modifications at once, and that it has low toxicity. There are a number of improvements that have been achieved over the last 6 years, and this is really with regard to the specificity of Cas9, with enhanced version of the enzyme that has been developed, and also a delivery system that have been developed according to the CRISPR-Cas9 technology to deliver, and also science going on to try to see how to favor homologous directed recombination over non-homologous enjoining repair mechanisms in certain types of cells. So as I said, very fast, it was shown to work very well in plants, in human cells, in different model organisms, also in fungi. Sure, it depends on the cells and organisms that you are studying and using. There are some cells that are more refractory because it's difficult to deliver the technology in certain types of cells, such as primary cells of patients. And surely as well, it depends, the success of the technology depends also of the natural repair mechanisms that will be triggered. So it's always important to have in mind, because as much as the general, let's say, frame to use to apply the technology is similar, one always has to come up with really understanding exactly what you bring into the cells, how you will deliver the technology into the cells, how the RNA will be designed. So it's always going quite much in details after in the protocol that will be used, and to refine the protocols according to the needs. What is really extremely amazing by this natural mechanism, so an enzyme having two nuclease domains, an enzyme that is guided by and can be programmed by two RNA molecules, a duplex of two RNA molecules, is that it gave a very fast number of ideas how to harness in a very efficient manner to harness the CRISPR-Cas9 system into different versions of the technologies that will lead to the CRISPR-Cas9 toolbox. And only in 2014, there were already all those, here are just selected aspects of the different versions of the technology used. So the fact that Cas9 can be surely designed to introduce small indels into the genome in a sequence-specific manner, or to insert new pieces of DNA or replace DNA, to also lead to larger deletions and rearrangements on the chromosome. That if one would use a version of the enzyme that does not cleave any longer the DNA, one would design as such an RNA programmable dead Cas9, meaning an RNA programmable Cas9 that can bind the DNA but cannot cleave any longer the DNA, and use this specificity to study initiation of transcription, or to block initiation of transcription, and also that if one would fuse to this dead Cas9 an activator domain or inhibitory domain, one could activate transcription or inhibit transcription. One could also fuse to that Cas9 another type of effector domain that would allow to modify the DNA and study epigenetic marks, or one could also fuse the dead Cas9 to, for example, here GFP, that would allow to really mark the DNA in a sequence-specific manner. And so these are just some examples of how the technology evolved, and the technology evolves further by combining CRISPR-Cas9 technologies to other types of genetic technologies that were developed prior to CRISPR-Cas9 and also the latest years. Here is just to show you, and you know this obviously, that the number of the number of publications about CRISPR surely increased tremendously. So mostly with regard to applications, but also with regard to the biology of the system. And surely technologies that has been quite much democratized now in academia, and allows to really ask novel questions in biology. And the reasons for this, as I said, is the fact that compared to talon nucleases and zinc finger nucleases, the CRISPR-Cas9 nuclease system is easy to use and quite versatile. So there are a number of applications of the system in life sciences first. So for sure it allows to provide a technology that facilitates the understanding of functions of genes, for understanding biology in health and disease, but also beyond, to rewire biological pathways, and to also create better models for human diseases. So what I like to say is that CRISPR-Cas9 comes at a very nice time in terms of technology, whereby it can benefit from other types of technologies, such as deep sequencing of the DNA, high throughput sequencing of DNA, also this ability to perform screens, also, and all the robotics behind, also the tremendous efforts over the last 10, 20 years with regard to delivery technologies in different types of cells and organisms, and also in terms of growing different types of cells, whether it's stem cells or other types of human primary cells, and also imaging technologies so all these technologies brought together really give more sense to CRISPR-Cas9 technology. For sure, in the field of drug development, it's important because it allows to screen for new targets for therapeutics, so allowing better to engineer better models to treat, to test medicine that are under development. It's also critical for example, cancer immunotherapy with regard to the engineering of CAR T cells that are meant to recognize cancer cells of patients and destroy them. And with regard to the agriculture field, it's important for the engineering of plant crops, but the first application actually of CRISPR-Cas9 was really on these phage-resistant probiotics. So this was the purpose of the groups of Sylvain Moineau and Philippe Horvath, first to understand the CRISPR-Cas systems, and they were working with a strain of Streptococcus thermophilus that contained 3 different types of CRISPR-Cas, but where their idea was to use this S. thermophilus strain having different CRISPR-Cas systems in nature to challenge these bacteria with different phages and select for strains that will become resistant to all phages. And these strains have been patented, and they are used for for yogurt productions. Having said this, it's also very important for the engineering of industrial microorganisms for different functions of production of enzymes or for biofuel applications. So with regard to CRISPR-Cas9 in agriculture, surely CRISPR-Cas9 can edit genes in major crops. It's a faster and easier way to introduce desirable traits, and it's more precise than the traditional breeding or the use of chemicals or the use of technologies prior to CRISPR-Cas9 that were always involving the introduction of a resistance marker or other types of markers. And then surely the idea that CRISPR-Cas9 engineered plants would be labeled differently than genetically modified organisms, but obviously in the US there is this understanding that one needs to differentiate between CRISPR-Cas9 engineered plant crops and plant crops that are engineered in using other types of technologies, but lately the European Court of Justice has decided to actually that CRISPR-Cas9 engineered plants will undergo the same regulations than genetically modified organisms. So that is a little bit slowing down the production of plant crops in Europe in the future for commercialization purposes. With regard to medicine, there is an important aspect of CRISPR-Cas9 for the development of CRISPR-Cas9 as a direct therapeutics, specifically to treat genetic disorders, whereby first approach is to have a proof of concept whereby CRISPR-Cas9 will show success in combination of gene editing by CRISPR-Cas9 and cell therapy. And so it's what we call the ex vivo therapy, whereby the cells are removed from the patient, corrected in the lab using the CRISPR-Cas9 technology, for sure undergoing all the process of quality control, and reinfused back into the patient once they have been corrected from the mutation. So you have different clinical trials that have started already, specifically targeting blood disorders, whereby the cells of the patient can be collected more easily than other types of genetic disorders. So hopefully, in a couple of years, there will be some examples of success, at least for an ex vivo therapy. What is important to mention is that the CRISPR-Cas9 system has largely evolved in nature. One should not think that there is a Cas9 protein having exactly the same sequence of amino acid throughout the entire world of bacteria. Actually, the CRISPR-Cas9 system is mostly found in bacteria. There was only one archaeal species having been found to carry a CRISPR-Cas9-like system. But in contrast to other types of CRISPR-Cas system, it's mostly found in bacteria and nearly solely found in bacteria. But when we started to understand that there was a tracer RNA involved in this system, so that it was a system with two RNAs, the first experiment we did that showed that indeed there was an interaction, we looked back at the sequence, at this possibility of interactions, and also going further and looking at whether other bacterial species other than Streptococcus pyogenes would have tracer RNA. When you work on small RNAs or when you work on another type of molecules, if you want to have a big discovery, you always try to see whether your protein or your RNA that you could identify being new, whether it's universal, so it's present across different species and domains of life, or whether it's really specific to your bacterial species. And at the beginning, we thought that tracer RNA was really specific to S. pyogenes, until we figured out that the specificity of this class of molecule is that it has an anti-repeat that can best pair with a repeat of the CRISPR RNA. But for sure, the repeats of CRISPR RNAs are always different for the Cas9 system when you compare the repeats of this system in different bacterial species. So what we found is that we could find this Cas9 system in more than 350 bacterial species, and that the repeat sequences were always different. So what we did is we tried to find an anti-repeat sequence for each of the systems, and we found indeed, these are just a few represented here, that there was always an anti-repeat sequence that will define the tracer RNA. But outside this anti-repeat, the sequence of this tracer RNA are extremely diverse. So in a way, it's a universal type of molecule associated to CRISPR-Cas9 systems, but having always different sequences. What we did is we did a phylogenetic analysis also showing that the Cas9 proteins are extremely diverse. So roughly they have less than 1% amino acid identity if you compare the different Cas9 with one another. The specificity is really an arginine-rich motif that is important for Cas9, and the nuclease domains that are conserved. The rest is extremely variable, so it's quite peculiar. Having said this, you can classify this different CRISPR-Cas9 system based on the amino acid sequence of the Cas9 proteins, based also on the way the two RNAs interact with one another, so allowing to classify these systems. What is really also remarkable in the evolution of the system in bacteria is that you have long Cas9 and shorter Cas9, which allowed also to show, at least at the time when we did the study, that Francisella novicida, the longest Cas9, and Campylobacter jejuni has a shorter Cas9. The reason why I say this, it's for delivery purposes, to perform genetics in higher organisms or even in smaller organisms like bacteria. You always try to have a system that is as small as possible, genetically speaking, also to be able to deliver or transfer or electroporate cells. And remarkable also for us, because we were focusing on Streptococcus pyogenes, is that Cas9 from Streptococcus pyogenes is particularly efficient. Us and others have tried other variants of Cas9 and have found that it's not working as well than the Cas9 of S. pyogenes. So this was our chance to really focus on this bacterium. Here I just want to show a further diversity of all these CRISPR-Cas systems. So we were ultimately working on Francisella novicida because we wanted to understand also how one could how one bacterium could have long Cas9s and others shorter Cas9, also for the purpose of engineering further CRISPR-Cas9 system to be shorter and to carry on with the structural biology analysis of these proteins. But in 2015, actually, the group of Evgeny Konin with Kira Makarova identified other types of systems. So all these systems here came up in 2015. Whereby it was shown that you had other CRISPR-Cas systems with only one single protein for the stage of interference. So we focused on a protein that is called CPF1, now called Cas12a. And here is just an example to show that what we showed for this protein is that this protein is sufficient to carry on the function of processing of the RNAs and then recognizing and cleaving the DNA at the stage of interference. So in this case, you have only one CRISPR-associated protein and the CRISPR RNAs. You don't have tracer RNA. But this protein has two nuclease domains. So one RNase domain that allows to cleave the long precursor CRISPR RNA molecule into mature CRISPR RNAs, to which the Cas12a CPF1 enzyme remains bound, and ultimately will use the nuclease domain, the DNase domain, to then cleave the DNA upon recognition by the CRISPR RNA. And what is also, just to show you the diversity of the system, this enzyme in this case does not cleave in a blunt fashion, but cleaves by giving 5 nucleotide 5' overhangs. So this is just one example. Other colleagues found and studied the role of this enzyme from type 5 and type 6 system. So the group of Jennifer Derna and Fang Zhang, and has found that some target RNA molecules. So this is the case of C2C2 protein that can cleave RNA targets. So in this case, it's not the DNA that is recognized, but the RNA. So a lot of research has been done with regard to the biology of the system. The last 3-4 years have been very important with regard to the publication of papers that allow to also understand how the step of recognition is working. There are more CRISPR-Cas systems to be discovered, hopefully more defense systems that will be discovered in the future, even though it's not always easy to detect them. And that may lead to interesting technologies. And I would like to stop here, so I already went to the acknowledgement. I need to cite ERS Genomics and CRISPR Therapeutics, two biotech companies that I have co-founded with Sean Foy and Rodger Novak. I need to cite this for conflict of interest. And this is the picture of my lab. This was actually 1.5 years ago. And this is the nice building of La Charité. So my institute is based on the campus La Charité Mitte in the middle of Berlin. And these are very nice old buildings. And I would like to thank you for your attention.
Okay, thank you very much for your talk. And are there any questions?
Thanks for gifting us this amazing technique. As we know, it's very important for the microbes, but why does this system present in this microbial world in a very uneven manner? Because many, not so many bacteria have them, right? So
yes,
but
yes.
Yeah.
So first of all, you have to thank the bacteria for having this amazing system. I think it's just a question of evolution. You know, those systems have emerged because of the very strong arms race between bacteria and mobile genetic elements and archaea and mobile genetic elements in nature. And you have a large diversity in the microbial world and an interest for also the bacteria and also the viruses that have actually found ways to counteract these defense systems, surely. It's an advantage for the macroorganisms to have a large diversity of those defense mechanisms, including CRISPR-Cas, to always have different types of strategies depending on the threat or depending on the environment. Otherwise, it would be, I guess, too unified. And you know, it's not an interest for a species to disappear. So you have to have a large evolution to allow always survival of all those species and having, how do you say, a very modulable strategy through diversity to be able to always counteract or act differently depending on the environment and the changes in the environment.
Thanks.
And also the fact that it's found only in 50% of the bacteria, it's because you have other types of immune systems, you know. And also because those CRISPR-Cas systems originally, they were, and this is the hypothesis, these Cas proteins are also called Casposons, because it is hypothesized that they originate from mobile genetic elements, transposons, which will make sense, that the mobile genetic elements were first, and then there was an evolution to counteract them. And so the fact as well that those CRISPR systems have evolved over time, and they have also spread through mobile genetic elements. So having said this explains as well that they are not present in the entire population. And the reason why they are more present in archaea, it's because archaea is needs more, really a super defense against all the viruses and phages that they found, that they have to deal with in very drastic environments in which they live.
Hi. You mentioned that there have been developed some super resistant strains of bacteria. So I'm worried a bit about the phages. So the phages themselves, why they don't rearrange their DNA to escape the memory? I mean, how much of memory the bacteria could acquire, and the phages themselves, why they don't evolve to escape the memory. For example, the DNA targeted, it's so essential to them, it's like for the capsid or I don't know, some structural part. You understand what I'm saying?
Yes.
Why they don't rearrange the DNA themselves? I mean, once, if I was a phage and I know that I will not escape, right? The offspring, if something changes, maybe it will escape.
Yes.
And how many of these transposon elements can be put in tandem?
Yes.
For example, and also the last question, if a first element here can become obsolete later. So if the green could be replaced by a turquoise color, because I don't need it any longer, for example, as bacterium now.
So I will try to give some insights that will allow to hopefully answer the different questions you had. First of all, one could ask the question, how come is it possible first that the phage can inject its DNA and can be memorized, specifically if it's a lytic phage. You would imagine that the phage will kill the bacterium and the bacterium will not have time to memorize the phage infection. So what is thought is that the memorization is done when the phage is in a stage of being deficient. So it's what we call abortive infection. Meaning the phage can inject its DNA, but nothing much will happen. And this memorization event actually is not as efficient as interference. If you have really, and we could show it for the type 2 system, and others could show it for other types of CRISPR-Cas systems. If you have the machinery in place, this step will be very efficient to recognize the phage upon a second infection and kill. This stage is not as efficient. That is why it has always been difficult to study this step of memorization in vivo in bacteria. So far, there is only Streptococcus thermophilus as really a bacterium whereby one can show naturally in the lab memorization. For other types of bacteria, you really struggle. You don't see anything happening really in the lab. So having said this, with regard to Streptococcus thermophilus, so this mechanism has really been shown, and actually people eating yogurts eat yogurts now, I mean, with strains of lactobacilli and streptococci having these CRISPR systems, because this was a way to produce in a natural fashion bacteria that will be resistant to phages encountered during yogurt production. So what they did is that they took strains that had the CRISPR-Cas systems, and you can have 3 to 4 and even more CRISPR-Cas system in one bacterial strain, naturally speaking. And if you challenge the bacterium having these CRISPR systems with different types of phages, you would select for the strains that have become resistant through memorization. So the memorization can happen, so in S. thermophilus at least it can happen in the lab, which is good because this was used for yogurt production. You can have, and this has been described by sequencing of those CRISPR arrays, you can have more than 500 repeats and spacers, whereby I totally agree with you, the idea, and that's why mostly the memorization occurs by introducing a piece of DNA of the phage at the level of the leader sequence, because this for sure will be, this is the idea, will be transcribed more than the spacers that are in the 3' end of the CRISPR RNA. And this is an advantage for the bacteria to produce more of those RNAs that recognize phages, which they encountered relatively recently, because the spacers that are located more at the 3' end corresponds to mobile genetic elements which they encountered a long time ago and which they may not, have to encounter any longer because they are, with evolution, they may have disappeared. So this is the idea. Mm-hmm.
Is there a possibility that the bacteria faces the effect of targets as well?
Yes, so that the phages counteract the attack by the CRISPR systems, you mean?
Yeah, but I mean, when the guide RNA or RNA is expressed, if Cas9 could cut as well the bacterial.
It's to differentiate self between non-self. So in principle, yes, because the question here is that Cas9 can cleave at this level, so why not also cleaving the DNA? So first of all, if, and this it's important to say, if you were cleaving the DNA, you will not realize it because when you cleave the DNA in bacteria, repair mechanisms are very weak. So in principle, it's the dead end for the bacteria, so you will not see it really. But for sure, to differentiate between the self and non-self, it's mainly by the PAM sequence. So the PAM sequence, which are shown, is a stretch of NGG that is present downstream of the sequence that is targeted, is present on the phage DNA. And surely it's important for the recognition of the phage DNA because this will be essential for the recognition at the stage of interference. But this PAM sequence is absent here because it's replaced by the repeats. So this is a way for the system to not be able to target the DNA of the cognate piece of the CRISPR RNA. So this is a way to differentiate between self and non-self. And it's complex because the PAM sequence is not, for example, type 1 have PAM sequences, type 3 don't have PAM sequences. So you have really a diversity also of mechanisms among the different CRISPR-Cas types.
Mm-hmm. Sorry. My question is a bit different. I would like to know what is your viewpoint on the ethics of using CRISPR and in the realm of what just happened in China and so on. So if you can comment on it a bit. Mm-hmm. If not, I can understand.
No, I can comment on it without any problem. So first of all, the question of ethics with regard to CRISPR-Cas9 are not new. This relates to questions that were already raised in the 70s with the ability to recombine DNA. Now, having said this, it's not new neither because with the zinc finger nucleases that were starting to be applied in human cells at the beginning of 2000, so more than 10 years ago, and the talent nucleases that were more recent. I mean, talent nucleases did not have really the time to be applied, but zinc finger nucleases already in the mid-2000s, there were scientists already starting to use the zinc finger nucleases to edit the genomes of human cells. So those questions are not new. Having said this, surely CRISPR-Cas9 is broadly used now, so and it's also a technology that is described as being more efficient. But it's good in a way because it allows to really reconsider what we call transgenic. I think with regard to the ethics, it's certainly a technology that benefit the field of agriculture, whereby you have now the possibility to produce plant crops in a more precise fashion, at least with a more clean outcome in terms of sequences than the technologies prior to CRISPR-Cas9. For research, it's for sure a very useful technology that facilitates the genetics and allows to really answer biological questions. What I like, and I guess the ethics is anyway very well controlled with regard to research in laboratories. I mean, me working on bacteria, we are maybe undergoing more I don't know, for me, the word ethics, it's what I have been hearing on day one when I entered the microbiology lab with regard to always being careful to not engineer a bacterial clinical isolate that will have an antibiotic resistance marker that will not exist in nature. So for me, it's kind of natural to think of ethics, but I think now everything is well controlled with regard to the study of model organisms, so CRISPR-Cas9 is very useful. And in terms of for sure studying mechanisms in human cells, it's very nice because it's a technology that allows to study better a large diversity of cells, specifically human cells, human primary cells. So it comes from evolution and it allows to study better evolution. The only issue with ethics that I see, I mean, in the plant field, you could argue that it could lead to, any farmer to be able to produce a plant and having a lot of plants produced whereby there will be difficult to control every plant because you would have to sequence the DNA of every plant. But it's more a question of what kind of traits you want to bring to the plants rather than the technology. Then after, beyond research, it's for sure the issue of the technology used for designer babies, whereby my point of view is very strict with this regard. So at the beginning, actually, I was more, let's say, even more strict with regard to the use of the technology for editing the genome of human embryonic cells until I got convinced that with regard to the cells that are then used, I mean the cells, the human embryonic cells that could be used for research purposes, these are cells that are issued from in vitro fertilization that will be discarded otherwise and that are really used in this case for research purposes under very strict conditions of growth of not more than 14 days. And in this case, when it's done on a very strong regulatory fashion, first of all, and second of all, that this will not be cells engineered for the research, but that are anyway existing will be discarded otherwise. I believe it makes sense because it allows to understand better the early mechanisms of life, and those cells exist. Now the issue is more with regard to the modification of the human germline. And here it does not seem that there is sense of using the technology to somehow edit the human germline for counteract certain development of certain diseases, because all the prenatal diagnostics, all those technologies in place allow this. The technology is anyway not ready to be specific and for the manipulation of the genome of human germlines, but it does not seem that clinically speaking it makes sense in any case. And then for sure, for human enhancement, it's my point of view is that it should be banned.
Hi there. Oh, I've got one more question concerning the CRISPR array. So I was wondering, you were talking a lot about the memorization process, but is there any process, deterministic in the way cells forget the DNA or forget the
so this it's difficult to say in the sense that what is thought is that some species will be lost at some point. Specifically if they are not needed any longer, I mean, but it's a mystery because there are some laboratory strains, for example, of Escherichia coli, that have these CRISPR arrays, and yet the CRISPR arrays are composed of sequences that correspond to mobile genetic elements that they would not encounter in the lab. So the question is why they keep the system, and there are hypotheses mentioning that maybe these Cas proteins could have repair functions at some point when the bacteria are very stressed, that they could take over some repair functions. What is interesting specifically with those strains that have these CRISPR arrays, even though these are laboratory strains for a long time, is like the strain of E. coli K12 that has a type 1 system. And the promoter of the Cas genes and the promoter of the CRISPR array is actually inhibited by a regulatory protein. So the Cas genes and the CRISPR RNA cannot be expressed. So the question is, why do they keep this? This also explains why the system was not found in the past, because you have had a number of scientists who were focusing specifically in the 50s, 60s, 70s on the biology of phages and bacteria. This was the only genetic that they could do in a way to understand really mechanisms until after it was possible to do it with mice and other types of model organisms. And people ask themselves, but why the CRISPR-Cas system was not discovered, like restriction modification system with all those screens that they were doing? And the explanation is because this CRISPR system was off. And that's why this what I like. It's because, then after in the 90s, scientists started to do more genetics with other types of bacteria, and it's really going through the, doing the biology with all these other bacteria that led to also the discovery of the function of CRISPR, and to go on with the biology.
Hi, so I don't know if you would be willing to comment on the state of the lawsuit between you and Jennifer Doudna versus Broad Institute. So it's my understanding that it's primarily a fight between first to patent versus first to invent. So why do you think you have more of a claim to this technology against, say, Dr. Zhang from Broad Institute?
Yes, I cannot comment so much in details, as you may imagine. No, I think if you wish, if you look at the history, at least of publications, it became clear that at the beginning of 2013, a number of publications came out showing that CRISPR-Cas9 was working in human cells, plants, and other types of model organisms. And this was at the same time than the publication of the paper from the Broad Institute. So everyone refers to the 2012 science paper as a paper that provides the guidelines explaining how researchers can use this technology and program it for their own purposes. Another point that I can mention is that the provisional patent application from our side, so University of Berkeley, University of Vienna, because it's complex, my student was mostly in Vienna when I was in Umeå, and the patent has also my name, because in Sweden, it's an interesting fact of the fact that the intellectual property does not belong to the institute but to the researcher. So basically, the provisional patent application was, originates from spring 2012, so beginning 2012, so quite early on. And how should I say this? So these are facts that after you can you can dig further, look at the dates, read, and make your own mind.
Can you shout, maybe?
the humans have the power to edit life in the labs on plants, on zebrafish, on Drosophila for research purposes. You know, it's very difficult actually. Unfortunately, it is possible that in the years to come, one will have to wait, but you will not be able to control the use of the technology for designer babies. Now the thing is that it's nevertheless for me science fiction. I mean the idea of human enhancement is one has to consider that a number of characteristics are caused by multiple genes. And here CRISPR-Cas9 will not be able to in a way fulfill maybe the dream of certain parents with regard to certain traits which they would like their kids to have. With regard to the question that you had, actually, it does not, this story does not disturb my nights. But you know, it's also because this mechanism, I mean, it's really based on the discovery of tracrRNA, having really understood that this enzyme works with 2 RNAs, etc. And let's put it that way, I did not see the description of the mechanism in other papers, and I knew as well the field to know that there was competition at some point, and yet the final mechanism was not understood, obviously. But I know that I understood it at least, and with my collaborators we came to this article. So, and without understanding the mechanism, you know, it's clear. I mean, when you see this, you're like, if you don't understand the mechanism, you don't know how to design the technology. Therefore, fundamental research is important in this case.
Okay, thank you again. Our time is over. So I would like to thank you for CRISPR-Cas and for the talk.